Good morning. I'm Premlata Bhatt, your biology faculty for NEET. Let us continue with our class. Previous class we have already discussed about the first part of principles of inheritance and variation. Now we are going to discuss about the next topics. That is, first we discussed about Mendel's law of purity of gametes, then law of dominance, that is the monohybrid cross. You remember, I guess? Okay. Now, our topics are incomplete dominance, co-dominance, multiple allelism, and dihybrid cross. Today, I want to discuss all these topics. But before that, let us continue with the MCQs we started in the previous class. Okay, few MCQs are there. Let us solve that. You please also solve with me. Okay, and at the end, you have to tell me how many questions you got it correct. Okay, yes. Let us begin with our questions first. So the seventh question we are dealing with now, that is a pea plant parent having violet colored flowers with unknown genotype was crossed with a plant having white colored flowers. Okay. And in the progeny, 50% were white. The genotypic constitution of the parent having violet colored flowers was. See what is given here. Violet colored flowers with unknown genotype was crossed with a plant having white colored flower. You know already. Violet color is dominant and white is recessive. If dominant is crossed with the recessive, okay, and you will get 50% white. Which cross is this? If you can recall, I have told you about the test cross, right? In test cross, we get 1:1 ratio. That is 50% dominant character and 50% recessive character. So directly you can go into this without solving all these things. Directly you remember we are dealing with the hybrid, okay, and you will get T. Okay, this is the example from Mendel's experiment. I can go into violet flower only, but try to understand this one so that you can easily solve this question, okay. So this is TT, capital T, small t, and this is small t, small t. This is the recessive character, right? This is recessive character. Okay, what they said here. So let us go into this only. That is, they said violet color, okay, and into white. So we got two gametes in the hybrid and one gamete, one type of gamete in the recessive parent, right? And we are getting here 50% violet and 50% is white. Are you getting my point? So what is the genotype? Whether it is homozygous or heterozygous, if both the alleles are similar alleles are there means you have to remember it is a homozygous. If dissimilar alleles are there, it is heterozygous. So what is our option here? Option number 3, that is heterozygous. Did you get that, children? Yes? I hope it's clear to you. Let us move on to the next question. Okay. If A represents the dominant gene. What is A? A is dominant. Okay. And small a represent recessive. Okay. And which of the following would be the most likely result of the first generation offspring when capital A, small a is crossed with the small a, small a? Again, they are asking about test cross only. You can see heterozygous condition and recessive one. So what they asked, which of the following would be most likely result in the first generation offspring when AA is crossed with the recessive one? You know the result. One is heterozygous dominant, another one is the homozygous recessive, right? So what is your answer? What all options are given? All will exhibit dominant phenotype, wrong. All will exhibit recessive phenotype, wrong. Dominant and recessive phenotype will be 50% each. That should be your answer. Okay, got it, children? Now, next one is, okay, which of the following traits in the garden pea plant would express itself only in a homozygous condition? Okay, be clear with the question what they're asking about, then only you try to answer, okay? Which of the following trait in the garden pea plant would express only in a homozygous condition? When it can express only in homozygous condition, when it is recessive. If it is dominant, it can be expressed in homozygous as well as heterozygous condition. Now they are asking only homozygous condition means it should be recessive character. So you know there are many 7 pairs of characters selected by Mendel for his study. 7 dominant and 7 recessive, right? Contrasting traits, right? So violet flower is dominant and green pod color. This is okay. Yellow seed color. Yellow seed color is dominant. Green pod color. Here there is a this one again, this is a dominant, okay. Then terminal flower position, this is recessive. Are you getting my point? Right? So what should be your answer? Which can be expressed only in homozygous condition, that is terminal flower position, because it is recessive. Recessiveness appears only in a homozygous condition. Is that clear? Yes. Next question. A cross between two heterozygous plant was performed and 120 offsprings were produced, in which 90 plants, how many tall? 90 tall, okay, and then what? 30. Let me write it in the ratio form, 30 dwarf. That means find out the genotype of their parents. So what is the ratio here, children? If you can use your maths skill, you can understand 31s are 30 and 33s are 90. So that is 3:1 ratio. Where you are getting 3:1 ratio? That is in a monohybrid cross phenotypic ratio. So you have to find out the genotype of their parents. Okay, a cross between two heterozygous plant. So you know very well we are talking about the F2 generation, right? So capital T small t into capital T small t, this is heterozygous condition. Then we get, let us use the Punnett square to predict the probability of the genotype of F2 generation. So capital T small t, capital T small t, here capital T, capital T, capital T small t, then capital T small t, small t, small t. Can you see? 3 are tall and 1 is dwarf, right? So 3 dominant and 1 recessive. So what is the phenotype? Sorry, what is the genotype of the parent? Parental genotype is capital T small t and capital T small t. So what is your option? Option number 2. Is that clear, children? If it is clear, let us move on to the next question. Okay? Yes? Now next one, you have to solve it very fast. Maximum you have to take only 30 seconds, okay? Yes, in pea plants, yellow seeds are dominant to green. Okay, you know very well, yellow seeds are dominant, capital Y, and green seeds are recessive, small y, small y. Now, if heterozygous yellow seeded plant is crossed with a green seeded plant, that means heterozygous is capital Y, small y, is crossed with a green seeded plant. Then, what is the ratio of yellow and green plants would you expect in F1 generation? Okay, so if it is crossed, then what you will expect? What is the possible? They are asking about ratio of yellow and green plants. They are asking. Okay, so you know very well here. If I am using capital Y here and small y here, capital Y, then small y, then capital Y, small y. So how many of them are yellow and how many of them are green seeded plants? Can you tell me how many? Say 2 green and 2 yellow. That means 2:2. That means 1:1. Right? What is the ratio you are going to get here? 50-50. Can you understand from here? 50-50. That means equal. equal number 50-50. Yes? Yes, children? Is that clear? Directly you can go into the solution. So I am just using this long cut method to make you to understand how you can reach it. Okay, but as you solve many questions like that, you can directly reach into the result. Okay, exact result. No need of worry about whether it will go wrong. Never. If you understand the concept, you will reach the correct answer. Okay, yes, quickly you can reach. Now, next one. Which term describes an individual having two different alleles for a gene? Yes, children. Which term describes an individual having two different alleles? Dissimilar alleles. That means it is heterozygous. Yes. Let us go into the next one. Now, we are going to discuss about the deviation from Mendel's principles. You know very well, from monohybrid cross, Mendel explained two principles of inheritance. One is law of dominance, another one is law of purity of gametes or law of segregation. Now, what dominance says? Dominance says that whenever there are different alleles present in a hybrid, then only one of the allele will express itself where the other one will be suppressed. The expressed factor is called as a dominant gene and suppressed factor is called as recessive gene. You know very well this one, right? So, but there are certain examples as the scientist, the geneticist, okay, as they continue the experiment with other plants, they reach to a conclusion that always these characters are not expressed as the Mendelian principle. Sometimes there will be blending of characters. Sometimes both the characters can appear. Sometimes more than two alleles are present in a gene. These all principles, okay, conclusion they reached after certain experiments. Let us see what happened next. Okay, yes, so we are going to understand. Before understanding incomplete dominance, you need to understand what is a locus and all. Okay, so when I talk about the gene where it is present, you have to understand. Okay, there is a term called as homologous chromosomes. You know very well, whenever fertilization occurs, okay, two gametes have to fuse. Both the gametes, one is male gamete, another one is female gamete. Male gamete comes from the father, that is called as paternal, and the female gamete comes from the mother, that is called as maternal. Okay, so both the gametes they are having the chromosomes. So these two chromosomes, that is maternal and paternal chromosomes, they are similar in size, similar in the gene composition, and similar in the height also, number of genes, position of the genes. These all are similar in a paternal copy and a maternal copy. Let me elaborate this. Okay, we are having 23 pairs of chromosomes, right? Chromosome 1. Chromosome 1. This is chromosome 1, you imagine. Okay, this is from father and this is from mother. Both are having a same height. Then position of the centromere also at the same place. You can see the length of the chromatids. You can see the both the arms that you can see. Okay, here the position of the genes also. If this is the gene responsible for the tallness, and here it can be tallness or dwarfness. That means it is for the character called as height. Okay, it can be similar or dissimilar. But the position of the gene is for a particular character. Okay, here what is a homologous chromosome? They are composed of one maternal chromosome and one paternal chromosome, and they have same size, length, loci and same genes but are not identical. Genes may be identical or may be dissimilar. That you know already, homozygous condition or heterozygous condition, that is also possible. Okay, now you have to remember what is a loci. Loci means many regions where genes are found. This I already discussed. Locus. Locus is the position of the gene in a chromosome. Okay, now they have different DNA sequences, so contain different alleles. That also you need to remember because this will be shown in the question. Okay, so you need to remember all these things. Okay, yes, this is one homologous pair and this is another homologous pair. You can understand, I guess, right? Homologous chromosome. There are two chromosomes identical to each other in the size, then the length and the loci. Yes, next. Now, in a pair of homologous chromosomes, one is inherited from a father, one from a mother. I already told. Okay, paternal homologue and maternal homologue. Now, here you can see the color, the genes representing the color. Both are recessive here. That means homozygous condition. Here, both are dominant. Again, homozygous condition. Now, if you observe it here, one is capital C, another one is small c. Okay, so there are different types of variations present between the homologous chromosomes also that you need to remember. Okay, yes, heterozygous condition also possible, homozygous condition also possible. Okay, fine. Now, there are two types of gene interaction. Now, what are genes? I said already, that is segment of DNA which carries the information for the synthesis of protein, right? So, here, if you see two chromosomes, that means homologous chromosomes. Okay, here position of genes are similar, I said. Suppose this particular location, we can see axial position of the flower, and here we can see the gene for a terminal position of the flower. How they are always, you can see they are, both are alleles of a particular gene, right? At the same position they are present. They are between the two homologous chromosomes, so we call them as intra-allelic, between two, in between the allele, same allele. Okay, in between factors of same gene. Okay, same gene or alleles. I should write, proper form is alleles of same gene. If there is interaction between alleles of the same gene, it is called as intra-allelic interaction. Okay, this interaction, because of this interaction, one example I can give you. See, this axial position of the flower is dominant over terminal position. You know already, right? If it is not able to completely dominate the terminal position, then there will be blending of character. Okay, that is observed in incomplete dominance. Then, if both are dominant, neither one of the allele is not able to completely dominate the other. Both are expressive in equal amount. Then we can see codominance. Then, Mendel said that there are two alleles of a gene. But when we studied the population as a whole, in a particular individual, that is the same thing as Mendel said. Any gene has two forms. Okay. But as we studied the population, we observed that there are more than two alleles for a character. Okay, so that is called as multiple allelism. These are all three interactions are due to intra-allelic interaction. These phenomenon, that is incomplete dominance, codominance and multiple allelism, are due to intra-allelic interaction. Is that clear, children? One more thing is there in between the genes, that is, here you see, suppose this is chromosome number 1, this is chromosome number 2. In chromosome number 1, we have observed there is a gene for expression of axial position and terminal position of the flower. Here you see, if it is for purple colored flower and white colored flower, okay, that is in the second chromosome. If this first chromosome gene, that is axial position of the flower, if it interacts with the gene for purple color of the flower and it influences the expression of purple color of the flower, then that interaction is called as interallelic. That means between two genes. Okay, alleles of two genes. Interaction between the alleles of two genes, there is a type of interaction. Yes, that shows two phenomenon, that is polygenic inheritance and epistasis. This we will learn in the later classes. Okay, now let us start with the first concept, that is incomplete dominance. Okay, this is the thing I already told. So we are going into the topic incomplete dominance. See, this is studied by Carl Correns. Now you can recall what I said in the previous class. I said the Mendel's findings are rediscovered by three scientists. Yes, you know Teschermark, Carl Correns. Okay, these all the three scientists you know. So here, Carl Correns, the same scientist Carl Correns discovered that there is a flower called as 4 o'clock plant. Okay, so it is also called as Mirabilis jalapa, scientific name. And one more flower, that is dog flower. It is also called as Antirrhinum majus. Antirrhinum majus, that is the snapdragon or dog flower. It also shows the phenomenon called as incomplete dominance, or Mirabilis jalapa, that is the 4 o'clock plant. That also shows incomplete dominance. You know from the word itself, decode the word, then you can understand the meaning. Incomplete dominance. So the dominant allele is not able to completely suppress the recessive allele. You need to remember, okay? Yes, children. See, that means if something is not completely suppressed, it is also going to express itself. Both the characters will blend. So there is a blending of character. Blending of character. Are you getting my point here? See, these all points you will get in the question also, okay? The hints they will give in the question. Blending of character, then mixing of character. These are the terms given, mixing of character. Then next, they can give you average character, or they can give you in between character. If these characters are expressed in the F1 generation, that means this particular plant or the organism is showing the phenomenon of incomplete dominance. Is that clear, children? So what is the incomplete dominance? In a hybrid, okay, if the factors are not able to, the dominant factor is not able to completely suppress the recessive character, then there will be a hybrid which do not resemble either of the parents. Either of the parents, it will not resemble. Okay, it will not be similar to the parents, but it will be a new character arising here in the F1 generation. That is in between the two parental character. For example, if there is a red and white as parental character, so in the progeny, you will get pink. Okay, that is in case of flower color in a Mirabilis jalapa or Antirrhinum majus. Very important names, children. Okay, so where all incomplete dominance is observed, that is in the Mirabilis jalapa and Antirrhinum majus. Clear, children? Now next one. Okay, we are going to here we will represent this cross. Okay, so you can see here the diagrammatic representation is given, but I will write it how you have to just show the cross. Okay, first one, parent is, P is for parent, parent is red into white. You know they are pure breeding varieties. Okay, now what is the genotype? Genotype is capital R and small r smaller. What is the gamete? Gamete, only one type of gamete from each parent. Okay, because they are in a homozygous condition. So F1 generation, first filial generation, you will get capital R smaller. That shows a blending of character. that is pink color. Hybrid means there will be pink color. Okay, now when we do the selfing F1, what we get? Let us use the Punnett square. Okay, so we get capital R into capital R small r. Okay, here gametes are capital R small r and capital R small r. Let us draw the Punnett square here. Let me show it here. Okay, so 4 squares we are using here. Capital R small r, capital R small r. So capital R, capital R, capital R small r, capital R small r, small r, small r. According to Mendel, what should be the ratio? Phenotypic ratio should be 3:1. But here, 1 red, okay, and 2 pink and 1 white. What is the ratio? That is 1:2:1. What is the genotypic ratio? 1 capital R, 2 capital R small r and 1 small r. Genotypic ratio also same, right? So isn't it the deviation from Mendelian principle? Yes, Mendelian principle says that there is a phenotypic ratio 3:1 and genotypic ratio 1:1. But in incomplete dominance, we can observe phenotypic ratio as well as genotypic ratio. Both are similar. Okay, they can give you the hint like this. Name the phenomenon in which phenotypic and genotypic ratio both are similar. Then you have to say incomplete dominance. Okay, snapdragon. Okay, yes, clear. Now, next we have to understand the another example for a incomplete dominance, that is shown by the pea seeds. Garden pea plant seeds are there, na? In them starch synthesis takes place. Okay, what takes place in the seed? Starch synthesis. In starch synthesis, starch grain will be showing incomplete dominance. Starch grain size, it varies in different plants. When we cross the bigger, that is capital B and small b, that means bigger size starch grain is crossed with a smaller size starch grain having plants. Okay, then we get a medium sized starch grain, that is capital B, small b. What you can understand from here? Isn't it the average character? One bigger size, one smaller size. So what we got as a result? We got the average medium character we got, right? So that means it is showing in heterozygous condition, it is showing a new character. That means the dominant allele is not able to completely suppress the recessive allele. That is the reason here. Okay, children. Now, this example you need to remember for exams and all, they used to ask, okay, competitive exams, they used to ask. Okay, fine, children. Now, next one. Before that, let me give some more examples. In human beings, there are curly hairs, you know, right? Curly hairs human beings are having, maybe having, okay, and I am having straight hair, right? The straight hair, some are having straight hair. If these two persons are crossed, okay, straight hair person and curly hair person, so the resulting individual will be having wavy hair. What will be hair? Wavy hair. The progeny will be having, father is having curly hair, mother is having straight hair, the resulting child will be having wavy hair. What is wavy hair here? It is a middle character. That means it is the average character. That means you can say incomplete dominance. What you can say? Incomplete dominance. Clear, children? And one more example is there in nature, that is shown by Andalusian fowl. Okay, this is a bird. It comes in two colors. Its feathers are in two colors. One bird's feather is black in color. Another bird's feather is white in color. Okay, when we cross these two birds, we get blue colored, blue feathered fowl. Okay, let me write here capital B, small b, small b. Here we get capital B, small b, heterozygous condition. We get a chicken or a fowl which is having blue feather. Okay, it is the intermediate character. Is it showing any parental character? No. It shows an entirely new character, which is because of blending of a character. Okay, incomplete dominance. I hope these four examples you got a clear picture of what is incomplete dominance. Neither of the character is expressed. One new character will be appearing in the heterozygous condition. Okay, now next we are discussing about the how this dominance can be explained. Okay, you know very well the genes are responsible for the synthesis of protein. That proteins are the enzymes responsible for the chemical reaction taking place in our body, or they can be responsible for a set of chemical reactions that leads into expression of certain characters in our body. For example, our skin color. Okay, melanin is responsible for the skin color. For the production of melanin, there are so many steps of reactions taking place. If the step is blocked in a certain stage, that will lead to serious conditions, metabolic conditions. We are going to discuss in the later part of this lesson. Okay, we call it as phenylketonuria. That we will discuss. Genetic disorder it is. Okay, now you have to remember, if the allele is normal, okay, it is not mutated. Mutation does not occur. It has not changed. Normal allele means it will produce proper enzyme for the execution of the reaction, and the product will be formed. We will get the same character as our parent. Clear, children? And if the allele is producing a non-functional enzyme, okay, it got mutated, you guess. Okay, mutation took place means there is a change in the sequence of a DNA. Then it will result into non-functional enzyme. Enzyme is not functioning means catalyst is not there. Then reaction will not occur, right? Reaction will not occur means character will not be expressed, right, children? Then sometimes there will be no enzyme produced. Okay, now here I said non-functional enzyme means the reaction may be happening but in a slow rate. That will be the reason for incomplete dominance. We have to study it further, then only we can understand. Right now you have to remember, okay, the normal gene means it will result into complete dominance. Non-functional enzyme means it will result into incomplete dominance, and no enzyme at all means recessive character. Recessive character. Recessive character is due to what? No enzyme, production of no enzyme. There is no production of enzyme. That means the genes which are representing that characters, they are not able to produce any enzyme. Okay, so that is the reason for a recessive trait, non-functional enzyme or no enzyme. That may be the reason for a recessiveness. This is again one question. Okay, yes. Now let us go into the question based on this incomplete dominance. Yes, children, are you ready to solve? Just start reading the question. A plant has 30 centimeter internode. You know very well what is internode. If this is the stem, there will be the nodes from where leaves arise. Okay, this is the node where leaves arise. This is one more node where leaf arises, or branches will appear here. Okay, now in between these two nodes, there is a gap. That gap is called as internode. What it is called as? Internode. The length of the internode decides the tallness or dwarfness. If the internode is smaller, the plant will be dwarf. If the internode is taller, plant will be tall. Okay, yes. Now, if 30 centimeter internode, suppose this is 30 centimeter, one more plant, this is how much is there? 15 centimeter. Suppose this internode is 15 centimeter. If these two are crossed, then What should be the length of internode if it shows incomplete dominance? 30, okay, and 15 in between. Divided by 2 you have to do. 30 plus 15 divided by 2. Average you have to take. So how much it will be? 45 divided by 2. What is it? 45 divided by 2. What is it? 2 2s are 4, 2 2s are 4, 2 5s are 10. 22.5 centimeter. Where is it? That is B. Clear, children? Average character. That is why I said. Okay. Now next. An animal breeder. Animal breeder crossed black birds with white birds. Remember, black birds are crossed with white birds. Okay. Now he got gray birds. What he got? Gray birds. Okay. Capital B, small b, that is gray. Now what he did? He crossed gray birds together. Okay. F1 generation. Selfing gray birds. Then he got 300 offsprings. What should be the approximate number of out of 300? You know very well. There should be 1:2:1 ratio, because this is showing the incomplete dominance. You can understand gray bird, right? 1:2:1. Let me show you the Punnett square if you are getting confused. Capital B, small b, capital B, small b. So here 1, capital B, small b, 2, capital B, small b, 1, small b, small b, right? So they are asking how many white offsprings. White is completely recessive, right? So here you need to remember only 25%. That is 25%, 50%, 25% in this ratio. So out of 300, what is 25%? Children, tell me. So 150 will be the gray bird, and 50, right? 75, sorry, 75 and 75, right? 75, 75 are the dominant characters. Can you see? Where is it? Option is B. Option B. 75 will be white offspring. 1:2:1 ratio. Out of 300, 75 white, okay, and out of 300, 75 black, okay, and 150 out of 300 will be gray in color. Okay, children? Yes, you can do it very fast. No issues. Now, next question. Phenotypic and genotypic ratio is similar in case of, just now I explained, so you can write it directly. That is incomplete dominance. Okay. Now, next one. Incomplete dominance is demonstrated in, what is the example given in the textbook? That you need to remember. Two examples are given. One is antirrhinum species or snapdragon, and another one is what? That is, you know very well, 4 o'clock plant, I said, Mirabilis jalapa. These are the two examples. So you can tick mark directly, antirrhinum species. Okay. Now, next one, we are going to discuss about the multiple allelism. The next concept we have to study codominance and multiple allelism. I said, Mendel told that one of the allele will express, another one will be suppressed in heterozygous condition. But in incomplete dominance, we observed that when the factors are in heterozygous condition, there will be a blending of character resulting in one new character, right? But there are certain examples where both the characters are equally expressed. Okay, neither of them are dominant. Okay, so here both the parents are expressed means the F1 generation resembles both the parents. How it is possible? Let us see one example here. For example, blood group system, ABO blood group system in human beings. You know, there are blood groups, blood group A, blood group B, blood group AB, and blood group O. For this, I have to explain you the concept of blood grouping system. On what basis blood is grouped into 4 groups? So let us understand that. So, in our blood, there is a cell called as a red blood cell, RBC, erythrocyte. Its shape is a biconcave shape. What is the shape? Biconcave, and it is not having any nucleus or mitochondria in it. You know very well. Its function is, it is having a hemoglobin, pigment called as hemoglobin. It carries oxygen. Yes, this much information you are already having. Now, this RBC will be having a plasma membrane. This is the outermost membrane. This membrane will be having a sugar polymer on its surface. What is there on its surface? Sugar polymer. This sugar polymer may be present. See, this RBC is having a sugar polymer A. Okay, so we are giving just a variable we are giving. Okay, another blood RBC, in another person's RBC, there is a sugar polymer called as B. Okay, and in another person's blood, RBC is carrying two sugars, sugar A and sugar B. Okay, this is AB blood group. So A sugar polymer is there means blood group A, B sugar polymer is there means blood group B, and A and B both are present means it is blood group AB. No polymer, sugar polymer is present, then it is blood group O. This much you know, right? In first year you have studied about this. Now, who is responsible for the production of these sugar polymers? That is I gene. Which gene is responsible? I gene is responsible. Okay, I gene is responsible. It comes in three forms. Okay, three alleles are there. Capital I, A, then I, capital B, then small I. These are the three alleles of the gene I. Can you understand how many alleles are there? Three alleles. Isn't it more than two? So, three alleles are present for I gene. So, this I gene shows multiple allelism. What it shows? Multiple allelism. Are you able to follow me? Yes, children? Okay. So, who is responsible for the formation of sugar polymer on the RBC surface? That is I gene. How many alleles are present in the I gene? Three alleles. Generally, how many are present? Only two alleles are present. Okay, there is a twist, I will explain that. Okay, so which are the alleles? IA, IB and I. These are the alleles. Okay, now is it present in the same person? No. When we study a population, human population, we will get three alleles for this I gene. Clear? Whatever Mendel told, he told in an organism. Okay, there are two alleles for a character. That you need to remember. Okay, so multiple allelism is shown by the I gene present in the human blood group. Right? Now, we have to understand the other things also. Okay, now I said this is the thing you need to remember. Okay, red blood cells have a sugar polymers. Okay, and its kind of sugar is controlled by the gene I. Okay, how many alleles are there? IA, IB and IE. Okay, there are more than two alleles. So, for governing a character, particular character, there are more than three alleles. So, we call this phenomenon as a multiple allelism. Multiple allelism is shown by ABO blood group system, right? And since in an individual only two alleles are present, multiple alleles can be found only when the population study is done. Okay, that you need to remember, children. Okay, now next, let us write the blood group, then antigen present and genotype. This will surely going to help you to solve the questions. Okay, blood group or phenotype. Blood group A. This is the phenotype, okay? And which antigen is present on the RBC surface? A. Sugar polymer, I said, no, it is also called as antigen. Antigen A. And what is the genotype? Which gene is responsible here? IAIA. This can be dominant or capital A, then a small I here. That means you need to remember here, small I is completely recessive. Okay, let me go back to the previous slide. so that I can explain you about this. Okay, here IA and IB both are dominant. What they are? Dominant alleles. Okay, dominant alleles. Okay, and small I, this is recessive allele. Okay, three alleles are there. Out of that, two alleles, IA and IB are dominant alleles. Small I is a recessive allele. So its presence will not affect the expression of the gene. You have to remember. Here you see capital IA or IAI. This is the genotype for blood group A, and blood group B. Which antigen is present? Capital B. And what is the genotype? Maybe homozygous or a heterozygous condition. Okay. This is the possible genotype for a blood group B. Homozygous B or heterozygous B. Homozygous A or heterozygous A. Now next, AB blood group. Which are the antigens present? A and B. And here the genotype is IA, IB. And which is the last group? That is blood group O. Which antigen is present? No antigen is present. Then what is the genotype? Small I. This much you need to remember, children. Will you remember? Now total phenotype. How many phenotypes are there from here? 1, 2, 3, 4. How many phenotypes? 4. How many genotypes are there? 1, 2, 3, 4, 5, 6. How many genotypes? 6. Remember this. 4 phenotypes are there in ABO blood group system and 6 genotypes are there. Clear, children? Any doubt? Now, next. In ABO blood group system, as I already told, it shows the phenomenon of dominance. If you observe IAI, this IA gene is completely dominant over the recessive gene I. Okay, and in IBI, capital B, that is antigen B is completely dominant over the recessive gene I. Now, this is called as a dominance. What it is called as? It shows dominance. Fine. Now, if IA and IB both are present, both are able to express equally, that means it is showing codominance. What it shows? Codominance. Right? Now, multiple allelism, because gene I has 3 alleles, IA, IB, I. So, which are the phenomenons showed by the ABO blood group system? It shows dominance, it shows codominance, it shows a multiple allelism. Okay, these are the points you have to remember. Next, one more example is there in our nature. If you observe the cattle, cow, okay, if there is a red cattle and a white cattle, if they are breeded together, there is a cross between them, fertilization occurs, then F1 hybrid, okay, it will be having a mixed color, that is red cattle with the white patches. Okay, that means here you can observe both the characters, that is if I am taking it as R small r and RW, okay, both the genes are able to express equally. That is how we are able to see both the characters expressed on the skin of the cattle. Okay, this is called a roan, red colored body with white patches you can observe. This is also one more example for a codominance. There is no blending of color, you need to remember. In codominance, both the characters are expressed without blending. Okay, yes. Now, next, we are moving on to the next concept, children, that is dihybrid cross. Dihybrid cross means two gene inheritance. In monohybrid cross, Mendel studied only the expression of one character at a time. Here he studied or observed the expression of or inheritance of two characters at a time. Two characters are governed by two genes, di, right? What is the word? Di means two. Two genes are present in a hybrid, that is dihybrid cross. Okay, now which all colors he has used, children? He used the seed shape and the seed color. Which are the characters he selected? Seed shape and seed color. Okay, you can take any two colors from the Mendel's 7 pairs of characters. Okay. Yes, seed shape. What is the seed shape which is dominant? You know, round shape is dominant over wrinkled seed, right? And uh, seed color, which is the seed color? Plus I need to write, not the into. Okay. What is the seed color? Seed color is yellow is dominant over green. Okay. Yes. Seed shape and seed colors are taken for Mendel's study. Which seed? Garden pea seed. Okay. Round seeded plant is crossed with wrinkled seeded plant. In the same plant, the seeds color, round seeded plants will be having yellow color in it. Wrinkled seeded plants will be having a green color in it. Such characters are selected by Mendel for studying dihybrid cross. Okay, so roundness is showed by capital R, and yellowness is shown by capital Y. Here small r, small y. These are the alleles we have chosen for our understanding. Okay, now this is genotype. Now gametes, whenever we are taking the gametes, the alleles for a particular character have to be present in the gamete. One allele for a particular character. See, for roundness, one allele. So capital R. For yellowness, one allele. So small r, capital Y. Here again, small r, small y. Did you understand, children? Both the characters will be having their particular alleles. One of the allele goes to the gamete. As there is homozygous condition, we will get only one type of allele here. Capital R, capital Y, small y. Is it confusing, children? I think it's very easy if you have studied monohybrid cross, right? So in the F1 hybrid, what we get? 4 alleles, capital R, capital Y, small y. Okay, here he observed all the seeds are round and yellow. How they are? Round and yellow. This is what he has observed. Clear, children? The F1 generation. This thing clear? Which plant he selected? The plant having the round and yellow seed and the plant having wrinkled and green seed. Okay. And the genotype is capital R, small, capital Y. Here small r, small y. Capital R, capital Y, small r, small y. These are the things you need to remember. Okay. Next, F2 generation. When these F1 generation are selfed, what happens? Let us see. This is the genotype, capital R, capital Y, small y into capital R, capital Y, small y. Okay, and let us form the gametes here. Each character will be having a one allele for the gamete, right? So capital R for roundness, capital Y for yellowness, then capital R for the roundness, small y for the green color, right? And small r for the wrinkled shape, capital Y for the yellow color, and small r, small y. This is a wrinkled shape and green colored seed. The same way here also, capital R. Let me show you in the circle itself. Okay, so capital R, capital R, capital Y, capital R, small y, then small r, capital Y, small r, small y. These are the gametes. Now we have to do the crossing. Okay, we should use the Punnett square. Let us use the Punnett square here. Okay, let me complete this. So the gametes we will write here, capital R. So ry, then here small r, capital Y, small r, small y. The here also the same thing, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the things, and you will get here, you have to cross multiply. Okay, y, capital R, small y. You know now how to do this, right? Very easy. And you have to write the phenotype also. Heterozygous condition means dominant character. You need to remember. Okay. Yes, very easy. Just write the phenotype. Okay. I'm writing the genotype here. So we'll fill in the next class, I guess. It's already time is running very fast. Okay. Let me complete this. Capital R, capital Y, small r, capital Y, capital R, small r, capital Y, small y, small r, okay, small r, capital Y, here, small r, small y, here, capital R, small r, capital Y, small y, capital R, small y, small r, small r, capital Y, small y, small r, small y. So here you will get dominant character 9 and 9 is to 3 is to 3 is to 1. This is the phenotypic ratio you need to remember. Okay, 9 round yellow. Round yellow, how many? Round yellow, 9. Okay, and then round green, you will get 3. Then wrinkled yellow, you will get 3. Okay, and there will be wrinkled green, recessive parental character. You will get only 25%. That means 1. What is the ratio? 9 is to 3 is to 3 is to 1. This is the ratio you are getting here, phenotypic ratio. Okay. Very easy to remember. 9 is to 3 is to 3 is to 1. Round yellow is the parental character. Wrinkled green again is the parental character, which is the new character you can see here. That is round green, which is not seen in the parental generation. Wrinkled yellow, this also not seen in the parental generation, but it appeared here in the F2 generation. You can understand here. So this phenomenon, why the expression of a new character appeared in the F2 generation, we will learn in the next class, and we will also discuss about so many questions based on a dihybrid cross. Once you revise the lesson up to dihybrid cross, we'll continue with the MCQs in the next class. Hope you understood the class. Thank you so much, children, and bye for today. Okay.